So let's consider the drivers or controllers of the biochemistry of the cell, enzymes. So this again is just a very basic introduction to enzymes. And we need to understand that because ultimately we have a better understanding of how genes work if we understand that all these proteins that are enzymes controlling the cell's biology are encoded in genes. So let's talk about enzymatic reactions. First, let's look at sucrose. So sucrose is a disaccharide. This is basically the sugar that you have on your table. And it can go through a hydrolysis reaction where you break it down to glucose and fructose. Now, this is a reaction that ultimately can lead to the release of energy as we use this and burn this up in cellular respiration. But this isn't necessarily going to happen quickly. And I can put sucrose into a glass of water and leave it there. And some of it over a great amount of time may break down into glucose and fructose, but generally it's pretty stable. So what we have to do, we can't just wait for the glucose and fructose to appear to feed into cellular respiration. We have to promote that reaction. So we illustrate that as a cartoon interacting with the enzyme sucrase. We see that we can make glucose and fructose by promoting the breakdown of sucrose. So what this is going to do is introduce some of the terminology that we're going to use with enzymes. So an enzyme, in most cases, are proteins. And we know that proteins work because of their shape. In this case, it's particularly obvious. So this protein has a weird shape. It has this kind of divot in it, you see on the left, that in which the substrate sucrose fits. So the enzyme has a substrate. That substrate is the thing that it binds to, that it interacts with. And that interaction is like a lock and a key. Right? So the surface of that protein is built in a way so it fits very, very specifically with its substrate, with the molecule that it's going to interact with, or molecules. So think about that as a lock and key, that pattern. Where does that pattern come from? It comes from the amino acids that make up the protein in the specific order that they're in their primary structure, folding into secondary and tertiary structure. Right? So that enzyme then is going to bind, in this case, sucrose as its substrate, and then promote the reaction. Water is going to come into that play, into what's called the active site of the enzyme. That's where it binds. So the active site of the enzyme on the protein is going to promote the chemical reaction, catalyze it, spit out then a glucose and a fructose, and be ready to roll to grab another sucrose. So that's the catalytic component. Because again, the definition of a catalyst is going to be something that facilitates the reaction without being consumed by it. So we can use it again and again and again. Think of like a little Pac-Man machine, chomp, 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 chomp. Each time it bites down on one of those little dots, it's like doing a reaction, right? So it's biting down sucrose, 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 sucrose. So it's much more efficient if I can build this tiny little machine to promote a chemical reaction and can keep going, right? I don't have to build a new one every time. So again, that fit, the shape of the protein is ultra important. So the shape of the protein is like a lock and key with its substrate. Now sometimes enzymes need helpers. And so we talk about micronutrients. This literally is the vitamins and minerals that you eat. So the proper name for this is called a cofactor. So in this case, you're showing vitamin C as a cofactor that binds into the active site of the enzyme to allow it to have the right shape so it can bind procollagen to make collagen, right? So in this case, just the amino acids aren't enough. You need another factor, a micronutrient, to bind to the protein to make it the right shape, because form and function needs to be just the right shape to work. So this idea that your vitamins and minerals, how they work, is interacting with enzymes to get them to work properly. We can also take that and do the opposite of it and talk about enzyme inhibitors. So this is how most drugs work. So when a pharmaceutical company makes a drug, or you go to herbal source for a medicine or what have you, it's the components, it's the biochemicals in that drug or that plant or what have you that one of them binds to the enzyme. In many cases, it may bind to the active site and then prevent the enzyme from binding to the substrate. So this is called enzyme inhibition. These are inhibitors. Why? Because it's the shape of that protein that's important. If I interfere with that lock and key, then it doesn't work. Or if we have what's called non-competitive inhibition, it binds to somewhere else on the enzyme and it changes the shape of the active site such that the substrate doesn't want to bind there anymore. So enzymes are everywhere. They're directing the chemistry of life. All right. Remember, they're proteins. And so a protein can get quote-unquote killed if other things are off. Temperature. Temperature is too hot, protein doesn't work. It unfolds. It loses its shape. Salt's too high or too low. Doesn't work. pH, it's too acidic. It's too basic. Doesn't work. So all these things affect proteins. So it's very, very important to maintain the right conditions inside a cell for life to continue on. So remember, this is everywhere. ATP synthase. This is an enzyme. What reaction is it catalyzing? It's catalyzing the reaction of adding a phosphate onto ADP to make ATP. All right, so that head group is just spinning around and around continually to like turning a water wheel and using the energy of the proton gradients to make ATP. Now we can also link enzymes together. So this is a case of quaternary structure, where we have a many-step pathway to make a particular biomacromolecule, in this case, a lipid, so fatty acid synthesis. So you can see here you've got different proteins, each as one of the different regions on this enzyme complex, and it will do one step of the process, hand off its the product to the next, and that product is the reactant for the next enzyme in the process, and it goes through the whole chain. It's like an assembly line to get out the final product at the end. This is increasing efficiency of the cell. So again, proteins are driving the chemistry of life. Proteins in the form of enzymes. Right? So it's super important. We're going to see then, with enzymes in the background, our last kind of component of our cell section is going to be how do cells reproduce. We want to remember that the very complex processes by which we see cell reproduction are all being driven by proteins.